I'll not give special quiz. Yes, she's stopping. From this time on, I'm not giving special short quizzes anymore. Okay? So our topic for today is on replication of DNA in RNA. So define DNA replication. So DNA replication is the synthesis of a replica. From the word replication, synthesis of a replica or identical copy of your DNA during cell division. So when cell will divide, then DNA replication will occur. What do you call the process of cell division? Mitosis. Okay, mitosis. So that's when DNA replication will occur, specifically in the S phase of the cell cycle. Okay, S phase of the cell cycle. So, what is semi-conservative replication? When we say semi-conservative replication, during the process, the two strands will unwind, and each strand will serve as a template. This is one template, this is another template. So each strand will serve as a template in the formation of a complementary strand. And therefore, the daughter DNA molecules that are produced consist of one parent strand and the other a strand that is complementary to the parent strand. So that's the meaning of semi-conservative replication. Each strand will serve as a template in the synthesis of a new strand, the one colored in purple, so that daughter DNA molecules consist of a parent strand colored blue and a new strand that is complementary to the parent strand. So that is the meaning of semi-conservative replication. So we'll now discuss the process of how DNA will undergo replication. Okay. So what are the three factors that are required in DNA replication? One, you need enzymes. Enzymes. What are these enzymes? So you have what we call the first enzyme, DNA polymerase. If it is a polymerase, therefore its catalytic activity will be to polymerize nucleotides. Okay? So when synthesis of DNA will occur, again, what's the building block of DNA molecules? Nucleotides. So therefore, if a DNA molecule will be synthesized, then its building blocks, nucleotides, will be polymerized, connected to one another. So the enzyme responsible for that is DNA polymerase. DNA polymerase. Now the other enzymes, you need also an enzyme we call DNA helicase. Helicase. DNA helicase. What is the catalytic activity of this enzyme? It will cause unwinding of the two strands in your DNA molecule, because we have said for DNA replication to occur, the two strands must unwind, and each strand will serve as a template in the formation of a new strand. So before DNA replication can occur, then it requires that the DNA double helix should unwind. And the enzyme doing that is DNA helicase. So you have a DNA helicase, you have a DNA polymerase. And the other one, you have what we call DNA topo, T-O-P-O, isomerases. Topo, T-O-P-O, isomerase. I-S-O-M-E-R-A-S-E. So what's the catalytic activity of these topo isomerases? If the two strands will unwind, then there will be a tendency for these strands that are separated to coil, to undergo supercoiling. So to prevent supercoiling of each strand that has unwound, then the catalytic activity of topoisomerase will be to prevent their coiling. Topoisomerases will prevent coiling of the two strands that have separated. Thus, the catalytic activity of topoisomerases. So how many enzymes now? 3. Another one. You have what we call a primase, another enzyme. A primase synthesizes the RNA primer. Okay? The primase synthesizes the RNA, ribonucleic acid primer. So later, we will discuss what is the role of this RNA primer during DNA replication. So you now have 4 enzymes. And the last is DNA ligase. DNA ligase. L-I-G-A-S-E. So what is the catalytic activity of DNA ligase? For example, in this strand, synthesis of a daughter strand or a complementary strand will occur in fragments. We call Okazaki fragments. This is named after the Japanese scientist who discovered that in the lagging strand, this strand, synthesis of your DNA will occur in fragments. We call Okazaki fragments. So if the nucleotide chains come in fragments, then they need to be connected to one another. And the enzyme causing breaks or nicks, okay? The enzyme now responsible for closing the nicks, the breaks in between nucleotide chains, it is your DNA ligase. So again, in one of the strands, synthesis of the complementary strand will come in fragments. So therefore, you have fragments of nucleotide chains, polynucleotide chains. And there are breaks or nicks in between. So these fragments need to be connected to one another. And the enzyme doing that is DNA ligase. So you now have 5 enzymes. Okay, DNA polymerase, a primase, a topoisomerase, a helicase, DNA ligase. Okay. So that's the first set of factors. The second, you need a DNA binding protein. So in this DNA that separate, you now have a single-stranded DNA, this one. This will act as a template. So what did we say earlier? This has a tendency to coil. So to prevent coiling, topoisomerase will exert its catalytic activity. And therefore, to stabilize the strand and to prevent it from coiling, the second factor will be a DNA binding protein. A DNA binding protein. The DNA binding protein will stabilize the template strand. So the third factor will be an RNA primer synthesized by your enzyme primase. Synthesized by your enzyme primase. So those are the three factors required in DNA replication. Enzymes, 5 of them. Second, a DNA binding protein that will stabilize its template strand. And the third, your RNA primer, Primer. So questions so far? So this is the primer. This one. You have two nucleotides here being shown acting as a primer found in the growing DNA strand. This primer, as I have said, is synthesized by your primase. And into this primer are added nucleotides. So this will grow as nucleotides will be added to this primer. So step 1, the unwinding of the double helix. So when will replication begin? After the double helix will unwind via the catalytic activity of your enzyme DNA helicase. DNA helicase. So as the double-stranded DNA will unwind, what will happen? A replication fork is created. So where is the location of this replication fork? It is found at the site where the two strands separate. The replication fork is found at the location where the two strands separate. So it's like you have a V line. So this is now what we call your replication fork. So the replication fork moves in this direction, going up. If this is the 5' end of this polynucleotide chain, then that is where the direction, this 5' end, that is where the replication fork will move. So what enzyme is involved in the unwinding? So we said DNA helicase. Sorry. Okay. Next, how does helicase cause the separation and unwinding of the double helix of your DNA molecule? Helicase will break the hydrogen bonds connecting the two polynucleotide chain. So helicase, it causes separation of the two strands. Angelo, please. Your helicase will cause separation and unwinding of the DNA double helix by breaking the hydrogen bonds that connect the bases. Okay, that connect the bases, or that connects the two polynucleotide chains to one another. So what is the effect and importance of the separation and unwinding of the double strand? So one effect, if the helicase will break the hydrogen bonds, it will now expose the bases in each of the strand. It will expose the bases in each strand. And therefore, each strand will now act as a template in the synthesis of a new strand. Why will the two separated strands become templates in the synthesis of a new strand? It's because formation of the new strand will depend on the sequence of the bases in the template strand. Remember the pairing of bases in the DNA molecule adenine pairs with thymine. If on this side you have the template strand, you have adenine, then the way a new strand will be formed will be a nucleotide thymine will be brought, okay, and become connected to your adenine base in the template strand. So that's why once the bases will be exposed, it makes possible the synthesis of a new strand using the bases as the template, because there is what we call complementary base pairing in DNA molecule. So that's the effect and the importance of the separation and unwinding. First effect, exposure of the bases, and then it becomes a template in the synthesis of a new strand. In what location? Question so far? Where are you now in anatomy and physiology? Who's your teacher in histology? Who's your teacher in anatomy and nurse? Are your teachers in nursing groups? she around? Oh, Sabella text her, she came but she didn't know. Maybe she was afraid that inside already. Tell her to come because we have short keys. Jopa. Again? Huh? CR? Our secretary saw her. She went down. Maybe she saw me and I was inside already. She was afraid. Tell her to come. It's okay with me.
Sir, you're so scary, that's why.
What?
You're so scary, that's why.
I'm not a scary person. I've been nice to you all the time. I'm just strict so that you will learn what you're supposed to learn. Sige, we'll continue. So in what occasion does separation and unwinding of the DNA double helix and replication occur? It occurs in many locations in the DNA strand. So you have here what we call origins of replication. These are the sites where separation and unwinding of DNA molecule will occur. It will not occur in only one portion of the double-stranded DNA. It will occur in several locations. And at that portions of the DNA double strand where separation of the strand will occur, a replication bubble is produced. These are what we call replication bubbles. So you have several sites where DNA replication will occur. And once a new strand is being synthesized, then they will be connected to one another because you have here breaks. And the one connecting the fragments of the polynucleotide chains, as we have said, is your enzyme DNA ligase. So what enzyme is involved in DNA replication that polymerizes nucleotides? It's your DNA polymerase. So we'll now discuss how a new polynucleotide chain will be synthesized by DNA polymerase. So where does DNA replication takes place? It takes place in each of the strand, in each of the template strand. So DNA replication takes place in each of the template strand. So in what direction does replication proceed? It proceeds from the 3' to the 5' end. So this DNA, if you can still recall, it runs in opposite direction. The two polynucleotide chains run anti-parallel. So if this is the first strand, then this is the 3' end going to the 5' end. So that is where replication of DNA, the direction will occur from the 3' to the 5' end in this template strand, while in this template strand it runs from here, 3' going to the 5' end, because the two strands run anti-parallel. Now, in what direction does the new polynucleotide chain grow? It will grow from the 5' to the 3' end. So do not be confused, because if a new strand will be formed from this template strand, so you will not be confused, then the direction of the growing of the new polynucleotide chain will be from the 5' to the 3' end, because if these two will now become a DNA molecule, then the two strands, always remember, they run in opposite direction. So that's why if replication will occur from the 3' to the 5' end, then the growing strand will be from the 5' to the 3' direction. That's the direction of the growth of the new strand. And please take note here, the replication fork moves from the 5' to the 3' end. So later it has an importance. So how does synthesis of no new nucleic acid strands proceed along the template strand? So what will happen? The DNA polymerase will read now the code found on the template strand. What is the code? The bases. Because what we said, formation of a new strand will depend on the bases found on the template strand. So it's like DNA polymerase will transcribe. It will read the bases found on the template strand, and it will bring nucleotide triphosphate based on the bases found on the template strand. So let's go back again. So how does synthesis of new nucleic acid strands proceed along the two template strands? This strand is what we call the leading strand. When we say leading strand, the synthesis of the polynucleotide will grow continuously. It will grow continuously. So this acts as the template strand, this one. And this is the growing strand that will be formed continuously in the leading strand. In the opposite strand, the new strand that will be formed is called the lagging strand. It lags, L-A-G-G-I-N-G. So when it lags, what will happen? Synthesis of a new strand will occur by fragments. By fragments we call Okazaki fragments. Okay, Okazaki fragments. So why is it that synthesis of the new strand of DNA in the lagging strand, why will it occur in fragments? So the explanation in the book is that it's because the synthesis of the lagging strand, the new strand, is going away the direction where the replication fork is being made. So this is where the direction of the movement of the replication fork, it moves going up. Okay? So synthesis will start from here, moving down. So it is going away from the direction where the fork is moving. So that's why synthesis of the new strand will occur in fragments compared to the leading strand, because synthesis of the new strand is along the direction where the replication fork moves. So that's why synthesis in the leading strand of the new polynucleotide chain will occur continuously. So explain why the leading strand. So we have discussed this already, right? It's because the lagging strand, the synthesis of the new strand is against the direction where the replication fork moves. That's why it's synthesized in fragments. While in the leading strand, the synthesis of the new strand is in the same direction where the replication fork moves. Sabella, did she reply? Juba. She's in the clinic. Meron pa siya? Leaky? Isa lagi? Kyrie? Okay, so maybe she's... Never mind. Tell her it's okay. Just secure a medical certificate. Okay, let's wait for Isabella to finish sending a message to Julia. So how are new DNA strands formed? So discuss the two central requirements in the synthesis of new DNA strand. One, it requires a template. So each strand in the DNA molecule will act as a template. This is one template. And the other requirement in the synthesis of a new strand is a primer. So this is the primer, to which will be added nucleotides. So nucleotides will be added one after another to your primer. So what will happen now? The DNA polymerase will read the bases in the template strand. So for example, this is the base, the first base, to which no new nucleotide will be added. So this base is adenine. So therefore, DNA polymerase will bring a nucleotide containing a base thymine. Okay? Because formation of the new DNA strand will be based on, is according to complementary pairing of bases. So it will be a nucleotide containing thymine as a base that will be added to this primer. And a hydrogen bonds will be formed between them, these two bases. While, so let's move to the other slide. So how are bonds formed during DNA replication? So this is the new nucleotide triphosphate. When nucleotides are being added to the primer, for example, if this is the primer, it is not a nucleotide. Okay, what did we say? Nucleotide? It is composed of a ribose sugar, one phosphoryl group, and one base. But during synthesis, it will be a nucleotide triphosphate. There will be 3 phosphoryl groups attached to the 5th carbon atom of the ribose sugar. So how will the incoming nucleotide become attached to the primer or to the growing end of a polynucleotide chain? So this is a ribose sugar in the primer or in the growing polynucleotide chain. So this is the 3rd carbon atom in the sugar ribose, to which is attached a hydroxyl group. The oxygen atom in the hydroxyl group will carry out a nucleophilic attack on this alpha phosphoanhydride bond. So because you have 3 phosphoryl groups, so the first bond is the alpha phosphoanhydride bond, and then this is your beta and gamma. So this alpha phosphoanhydride bond will become cleaved by this oxygen atom, and these two phosphoryl groups will be released in the form of a pyrophosphate. And once this is released, this oxygen atom now will share its electrons to this phosphorus atom. So this bond will be broken, this phosphorus atom. And in that way, the incoming nucleotide becomes attached to the ribose sugar of the primer. So the bond that will now be formed is what's the bond connecting nucleotides to one another? Okay, very good. Phosphodiester bond. A covalent bond will be formed between this oxygen atom and this phosphorus atom. So that's one ester bond. And this is another ester bond connecting this phosphoryl group to this ribose. So you now form a phosphodiester bond. And for example, if this is adenine, then this base should be thymine. And what will be formed between these two bases, adenine and thymine, will be hydrogen bonds. So the two bonds that will be formed as its nucleotide becomes attached to the primer will be your phosphodiester bond and your hydrogen bonds. So these are the bonds that are synthesized during DNA replication. So that is why the catalytic activity of DNA polymerase will be to polymerize several nucleotides. So step 3, closing the nicks, especially in the lagging strand, where synthesis of your new strand comes in fragments. So there are breaks or nicks in between. So what will happen now? They will be connected to one another, these fragments of nucleotide chains, via the catalytic activity of your enzyme. This nicks, this break, DNA ligase. DNA ligase. So here again, so you have new strands being formed, and this has to be connected with this new strand here. So that will be the catalytic activity of DNA ligase. So how long does replication take place in human cells? It will take several hours. It will take several hours. Because we have a long DNA strand, that's why it will take several hours. In fruit flies, your Drosophila, it will take less than 3 minutes only. Less than 3 minutes only. And why will it come so fast? It's so fast also in human beings, only that we have a long DNA strands. That's why it will take several hours. So it takes so fast the synthesis of the DNA molecule because replication will occur along several origins of replication in the DNA strand. So this is just a concept map on the structure of the DNA and DNA replication. Just take a picture and then read. We have discussed everything already. You can find this in Lippincott. I've given you Lippincott already. By the way, you tell Jova that if she cannot come on time, she is still welcome to attend the lecture so that she will not miss quizzes. But I think she really has to solve that problem. acids, RNA. So compare RNA with DNA based on the basis of ABCD. So we are going back because the next topic after this will be translation, okay, or transcription. The synthesis of mRNA using DNA as the template. So RNA, it is composed of one strand only. The base in RNA is your adenine, guanine, cytosine, and uracil. Compared to DNA where you have thymine. The sugar in RNA is ribose, where there is an oxygen atom in the second carbon atom of the ribose sugar, while there is no oxygen atom in the second carbon atom of the ribose sugar in DNA. And base pairing in DNA is adenine pairs with thymine, purine pyrimidine, guanine, which is a purine base, pairs with cytosine. But in RNA, adenine pairs with uracil, while guanine pairs with cytosine. So you have here guanine, cytosine, adenine, uracil, or if it is A here, then what should be found on this side should be uracil. Do not be confused with this one. This is tRNA. All RNAs, they are single-stranded, but they can form helical structures because they can fold back on itself. It can fold back on itself. So that in regions where there will be base pairing, then helices can be formed. So the primary structure of RNA is similar to DNA. This is RNA, but only that in RNA, thymine becomes replaced with uracil, while in DNA, you have thymine. Okay? So if base pairing will occur, then uracil pairs with adenine, guanine with cytosine, cytosine with guanine, adenine with uracil, or uracil with adenine. So the primary structure of RNA refers to the sequence of the nucleotides in the polynucleotide chain. And the sugar phosphate acts as the backbone on which are attached the bases. The bases are attached to the sugar and not to the phosphate group, although the backbone will be your sugar phosphate molecules. How many strands does it consist? RNA, it is composed only of one strand, but it can fold back on itself, and that's why helical regions can be formed. Where are helical regions formed? It will be formed on areas where there is base pairing. So in the bolts, you have no base pairing. In the internal loop, you have no base pairing. In the hairpin loop, you have no base pairing also. So therefore, base pairing can occur in regions other than the hairpin loop, the internal loop, and the bolts. So once there is base pairing, then this can form a helix. This is just tRNA, the secondary structure. The clover leaf representation, because the tRNA looks like a clover leaf, its shape. So this is the tertiary structure of tRNA, showing now the formation of helices or a helix. because it folds back on itself. So state the three types of RNAs. You have ribosomal, the site of protein synthesis. So it is a component of the ribosome, your ribosomal RNA. And then you have messenger RNA that carries the information from DNA to the ribosome. So messenger RNAs unformed, they are formed by a process we call transcription. Transcription. Again, the DNA will serve as a template in the synthesis of mRNA. And mRNA now dictates the synthesis of protein in the ribosome. Then your transfer RNA, this is what we call an adapter molecule. It brings specific amino acids to the ribosome that will be utilized now in the synthesis of protein. That's the role of tRNA. From the word transfer, bringing specific amino acid to the site of protein synthesis, your ribosome. So ribosomal RNA is the most abundant type of RNA. It is the most abundant type of RNA. Where is it found? It is found in the ribosomes, in the cytoplasm. What is it composed of? It is composed of, what is it composed of? RNA. tRNA, what is it composed of? tRNA again. What is it composed of? Louder? Louder. RNAs, whether T, mRNA, or rRNA, what are they composed of? So what is that? Okay, not good time. RNAs, whether mRNA, rRNA, tRNA, they are composed of nucleotides, just like DNA. DNA is composed also of nucleotides. So do not confuse R with ribose. Okay? All RNAs are composed of nucleotides. So the importance of rRNA is the site where protein synthesis will take place. Function of mRNA, it carries the information from the DNA molecule, and it is composed of codons, your mRNA. What are codons? Codons are triplets of bases. You have a sequence of 3 bases that will now specify the amino acid that will be formed, okay, that will be polymerized during protein synthesis. It dictates the sequence of the amino acid in the growing polypeptide chain in the ribosome. So where is mRNA synthesized? It is synthesized in the nucleus by a process we call transcription. Transcription. The one we have discussed earlier, that was replication, that will occur during cell division. But the importance of mRNA, when the cells will now need to synthesize proteins, that's where the role of mRNA will come into the picture. It will now be synthesized in the nucleus, and it will now dictate the synthesis of proteins in the ribosome. All types of proteins is the mRNA dictating their synthesis, whether it's hemoglobin, whether enzymes, whether transport proteins like albumin and so on. So again, it is composed of nucleotides. Next, function of tRNA. So we have said it already. It brings amino acids to the ribosome, the site of protein synthesis. Now there are two important sites in the tRNA molecule. What are these sites? The amino acid arm in the 3' end. This is to where the amino acid will bind, which tRNA will bring to the site of synthesis, your ribosome. This is to where the amino acid will bind. And it is this amino acid that this tRNA will bring to the site of protein synthesis in the ribosome. The other important site is your anticodon arm. What is the importance of this anticodon arm? This is composed of 3 bases that will be complementary to the codon in the mRNA. So how does the tRNA know? How does it know what amino acid to bring? It is via this anticodon. Because this is complementary to the codon, to the codon in the mRNA molecule. It is complementary. So that the tRNA will know what amino acid to bring. Each amino acid will be carried by a specific tRNA. If there are 20 amino acids, there will be 20 tRNAs carrying the different amino acids to the site of synthesis. So the anticodon arm, the amino acid arm. So this D arm and this TC arm, they are responsible in making possible tRNA to fold back on itself and form a helix. The D arm and the TC arm are responsible, making it possible for the tRNA to fold back on itself and form a helix. And the other importance of this arm, TC arm, this will enable tRNA to attach to the large ribosomal subunit. The ribosome is composed of a small and a large ribosomal subunit. So the TC arm will enable the tRNA to bind to the large ribosomal subunit. So we have discussed already the function of the two important sites. So this is just showing you the amino acid carried by tRNA on its amino acid arm, while this is the anticodon in the tRNA molecule that is complementary to the codon in the mRNA found attached in the ribosome. So this is a triplet. You have 3 bases, A, C, and U. Okay? So if it is A on the mRNA, what is found there on the anticodon? Uracil. C, guanine. U, adenine. So the anticodon will be here on the tRNA, U, G, A, while the codon in the mRNA is ACU. This ECU specifies what amino acid should be found in the sequence of the polynucleotide chain. So this will now be complementary to the anticodon. So that's how the tRNA will know which amino acid to carry to the site of synthesis. So this is a summary of the RNA. this one, the mRNA is the template utilized in the synthesis of protein, while the DNA is the template utilized during synthesis of a new DNA molecule during replication. And the DNA is also the template utilized in the synthesis of mRNA, while mRNA is the template utilized in the synthesis of a protein molecule. So next we move to the flow of genetic information. So what is the central dogma of molecular biology? The central dogma refers to the flow of genetic information. From the DNA, it is transferred to dividing cells by a process we call DNA replication during mitosis. Each strand of DNA will act as a template in the synthesis of a new strand. And that occurs during cell division. What about during protein synthesis? So the genetic information found in the genes in the DNA becomes transcribed, meaning the DNA will act as a template in the synthesis of an RNA molecule by a process we call transcription. So take note, during transcription, it is not only mRNA that are synthesized during transcription. All the types of RNA will undergo synthesis by a process we call transcription. Although what is highlighted in the discussion or in the figures during transcription, the one that is highlighted is the synthesis of mRNA. Although synthesis of rRNA and tRNA, they occur also in the nucleus in a process we call transcription. So all types of RNA undergo synthesis in a process we call transcription. And then once mRNA is synthesized, and plus you have the RNA and tRNA, then this will now dictate the synthesis of protein. In the process by which proteins are synthesized using mRNA as a template, we call it translation. So therefore, in the flow of genetic information from the DNA to the protein molecule, it consists of two processes, transcription and translation. While the flow of genetic information from the parent cell to daughter cell, it will just occur in one process we call replication. Replication. Summary, what we have discussed. What is good in Lippincott is that after it has discussed topics, then it will make a concept map. A concept map is a summary of what was discussed in a certain chapter or topic. It makes easier to remember everything that was taken up, for example, in a chapter. So we will not discuss any more transcription. You might be confused during our short quiz. So we'll take this up next meeting. So you have 15 minutes to prepare